Our today's topic is bacterial transformation. Although there is no sexual reproduction in bacteria, natural genetic transformation uh, and conjugation are regarded as parasexuality. Parasexuality means without fertilization. Both the process, that is uh, conjugation and transformation, mediate the transport of single-stranded DNA into the cytosol. However, conjugation is a plasmid encoded process and has been documented only in a very few species. For example, Escherichia coli and some Salmonella species. While bacterial transformation is widely distributed in nature and it doesn't depend on the presence of mobile or extra chromosomal genetic element. Bacterial transformation means uptake of DNA molecules through the cell wall from the external surroundings, followed by stable incorporation into recipient genome or replication as an independent plasmid, right? So it is one of the three horizontal gene transfer mechanisms found in bacterial cell. Three gene transfer mechanisms in bacteria are conjugation, transformation, transduction, right? Some bacterial species have evolved specific mechanisms. Competence for the uptake and recombination of external DNA. This sometimes involves the degradation of one strand of incoming DNA and incorporation of the other strand into the chromosome, right? Sometimes this uptake is dependent on the presence of high enough concentration of cell locally. This is called quorum sensing mechanism. What is this quorum sensing mechanism? Actually, it is a process of cell-to-cell communication that allows bacteria to share information about cell density and adjust gene exploration accordingly, right? In some cases, cells will only take up DNA that include a short specific sequence. Now, history of transformation. Transformation was first demonstrated in 1928 by British bacteriologist Frederick Griffith. Griffith discovered that a harmless strain of Streptococcus pneumoniae could be made virulent after being exposed to heat-killed virulent strains. What is virulent? Virulent means ability of an organism to infect host cell and cause disease, right? So Griffith hypothesized that some transforming principle, means from the heat-killed strain, was responsible for making the harmless strain virulent. But he couldn't explain the phenomena, and this was called Griffith's effect. In 1944, then, this transforming principle was explained by some other scientists like Oswald Avery, Colin MacLeod, or McLean McCarthy, etc. What they did, they isolated DNA from virulent strain of Streptococcus pneumoniae. And using this DNA, they were able to make a harmless strain virulent. They called this uptake transformation, right? Now, origin of this bacterial transformation. Griffith actually conducted a number of experiments with Streptococcus pneumoniae, a bacterium that causes pneumonia. He worked with two strains of this bacterium called rough, means R strain, and smooth, S strain. R strain is non-pathogenic, means doesn't cause any disease, but S strain is pathogenic and disease-causing. S strain also has a capsule outside its cell wall. In his experiments, first Griffith injected, you can see here in this diagram, living S strain into mice. And he observed that mice died from pneumonia. Then he injected a live R strain into mice. Mice survived. Then he injected mice with heat-killed S strain. Mice also survived. This showed that capsule, as we know, capsule is present outside the cell wall of S strain. So this showed that capsule alone was not the cause of death. Then he injected a mixture of heat-killed S strain and live R strain into mice. Mice died. Then Griffith isolated live bacteria from the dead mouse. Only S strain of bacteria was recovered. This isolated S strain was then Griffith injected into fresh mice. Again, mice died. Then Griffith concluded that something had passed from heat-killed S strain into live R strain and transformed it into pathogenic S strain. He called this the transforming principle, right? Now, another important term, that is competence in bacterial cell. What is this in bacterial transformation, right? So bacterial transformation may be referred to as a stable genetic change brought about by the uptake of naked DNA. Naked DNA means without associated proteins. And the competence means state of being able to take up exogenous DNA from the environment, right? There are two forms of competence in bacteria, natural and artificial. Natural competence, first we will discuss. In nature, only 1% of bacterial species are capable of naturally taking up DNA under laboratory conditions. Naturally competent bacteria carries sets of genes that provide the protein machinery to bring DNA across the cell membrane, right? Here, first, DNA binds to the surface of competent cell on a DNA receptor and then passes through the cytoplasmic membrane via DNA translocase. Only single-stranded DNA may pass through. One strand then, it means degraded by nucleases in this process. Translocated single-stranded DNA may then be integrated into bacterial chromosome by Rec-A dependent process. But in gram-negative cells, due to the presence of extramembrane, the DNA requires the presence of channel formed by secretins on the outer surface membrane. Artificial competence, it can be induced in laboratory procedure that involve making cell passively permeable to DNA, because some bacteria like E. coli are non-competent, right? So they can be induced under laboratory conditions by exposing to the conditions that do not normally occur in nature. Here, cells are incubated in a solution containing divalent cations. Divalent cations most commonly used are calcium chloride, right, under cold conditions, then exposed to pulse or heat shock. Here, surface of the bacteria such as E. coli is negatively charged due to the presence of phospholipids and lipopolysaccharides, right? And DNA also negatively charged. So one function of these divalent cations, therefore, would be to shield the charges by coordinating the phosphate groups and other negative charges. So allowing a DNA molecule to adhere to the cell surface, right? Second thing, it also weakens the cell membrane, making it more permeable to DNA. So heat pulse, what is the function of the heat pulse? Is thought to create a thermal imbalance on either side of the cell membrane, which force the DNA to enter the cells through either cell pores or the damaged cell wall. Now, what is the mechanism of bacterial transformation? Competent bacteria have DNA receptors on the surface of its cell. Free DNA, which is available in the vicinity of these bacteria, can bind to these DNA receptors. Double-stranded donor DNA binds to specific receptors on the surface of a competent cell, but only single strand of donor DNA reaches the interior of the recipient cell. Other strands gets degraded by nucleases, right? So then recombination with the recipient homologous DNA occurs and bacterial cell successfully transformed. Now, DNA uptake machinery, which is present on the competent on the surface of competent bacteria. A DNA uptake machine is assembled at the cell poles. So here in this figure, this is the uptake machinery of Streptococcus pneumoniae, right? So a transformation, actually what it comprised, it comprises a transformation pseudopilus, means a surface related to type 4 pili made of a major pillin-like proteins, COM-CG, and minor pillin-like proteins, COM-GD and COM-GE, COM-GG. Here you can see, right? So a single-stranded DNA receptor protein, COM-EA, then an endonuclease. Endonuclease identified in Streptococcus pneumoniae only. Means and A, here you can see pink color. A polytopic membrane protein considered as a transmembrane channel for single-stranded DNA, COM-EC, this, and an ATP binding protein, COM-FA, here, right? So, initial binding of exogenous DNA to the surface may involve interaction of negatively charged DNA backbone with positively charged residues in the pseudopilus itself. So first step is a surface endonuclease, the A introduces double-stranded cleavage into the bound DNA, right? Second step is disassembly of pseudopilus. It would then allow exogenous DNA to traverse the peptidoglycan, possibly driven by the proton motive force, thereby conveying double-stranded DNA into contact with COM-EA. And the third step is this double-stranded DNA receptor would then deliver this double-stranded DNA to end A to degrade one strand, right? The bacteria where nucleases is not there, then this DNA, double-stranded DNA receptor, deliver this double-stranded DNA to end A to degrade one strand. And the last step is permitting transport of its complement through the transmembrane channel, COM-EC, right? You can see here. This is the whole machinery, right? And passes off this. And this is in, means DNA in, and this is out, right? Inside. So this is all about transformation. Thank you.